So we've separated it out. So now we look at nuclear grade and pattern and the presence of necrosis, and we do them separately, so it's not just one package. Next. So this is just a little bit of higher magnification. You can see how some of these cells are on their way to die. They're not well preserved and healthy, or they're not healthy to begin with, but they're not well preserved anymore when they're here, and then the nuclei totally disappear, and there's all this necrotic debris.
Now, I'm going to go back.
Yeah.
See it at low power. This is ductal carcinoma in situ,
Right.
because Britt's going to get there in a moment, but you don't have cell, the basement membrane surrounds these ducts, and these cells are not breaking through.
It's all unbroken. You can't see any tumor cells outside the ducts themselves. So that's the definition of carcinoma in situ. So there's no invasive. And when you're at this stage, the tumor has not yet developed the capacity to metastasize. If it's all contained within the duct, it's still in situ. It's right there. It hasn't gone. Yeah.
All right, a question. You take a section through a particular part of the tissue,
Yeah.
and you see DCIS. How do you know further on down that duct that there isn't invasive?
You don't. It's a sampling.
Okay.
You know, pathology never looks at every cell in a tissue, no matter how. I mean, we could spend the entire U.S. health budget if we wanted to section everything at 5 microns. So we look at a sampling of what's there. And could there be a break in the membrane somewhere that we're not seeing? Yeah, there could be. There's a possibility.
But they don't just take one slide.
But we sample
They
pretty
sample
extensively.
multiple,
Yeah.
you know, so to
And
make
we'll get
sure.
to that part too.
That question comes up a lot among cancer
Sure.
patients.
Yeah, no, and
So
the
it's
truth
scary,
is
you
that,
know?
you know, it's all probabilities, is what it is. And that's true for margins, and we'll get to that too. None of this is black and white. And I sometimes joke and say, you know, when you look down the microscope, there's not a little sign down there saying what it is. You have to interpret what you see, and you do the best you can with this. All right, so we saw a high end of carcinoma in situ. Now this is the low end. So here the pattern is very different. We still have very dilated structures, but the cells are much smaller. The nuclei are small and round, and they look very similar to what benign cells should look like. But they're too many. It's too piled up, and the structures are dilated to a great extent. And then you have these round empty spaces fairly evenly distributed through this. So this is a low-grade carcinoma in situ with a cribriform pattern. The cribriform is this kind of pattern with empty spaces that are distributed throughout. So that's the other end of the spectrum of carcinoma in situ, but still contained within the basement membrane. Higher power view. And what I wanted to show with this one is how the cells are oriented. It's a sort of like a Roman bridge, kind of, where the cells are long edge from here to here. They're all pointing in toward the space in the center. And that's opposed to streaming when they're lined up in a different way. I'll show that later. So this is typical for something that is carcinoma in situ as opposed to hyperplasia. So next picture.
And the distinction is also that if hyperplasia, remember I said it wasn't clonal, and it's just an increased proliferation. Well, that doesn't generally have genetic changes yet. Whereas if you look at the DNA of DCIS, it does. So whatever we're seeing that Britt's eyes see, the distinction between DCIS and pre-DCIS, that's the point at which something becomes neoplastic with genetic changes, clonal versus not. So
and it's not always straightforward. It's sometimes very difficult to tell. There is a gray zone between the two, so it can be tricky to next. So here's an example of a very close-up view of high-grade DCIS. The basement membrane is still intact, running along here, and these are very large, very abnormal, very high-grade type cells, still carcinoma and such. Want to say something about that?
Well, you're going to get to grade later and
Yeah.
what makes a high-grade nucleus and so forth. So this is
just sort of getting used to the images a little bit before we get there. And here's yet another pattern. We call this a papillary pattern. So these are like little fronds sticking into the duct lumen. And so it's another way that carcinoma in situ can grow. But still you have the basement membrane intact all the way around. Okay, so we're back to the normal breast in cross-section, HER2 lobule, ductal, lobule, ductal. So we dealt with the carcinoma in situ that's in these structures, and now we're going to move to the lobules. We're going to talk about lobular carcinoma in situ, which is a slightly different animal. So lobular, as the name would indicate, originates in the lobules, not in the ductules or the ducts. It's associated with increased breast cancer risk. However, it doesn't happen where you find the lobular carcinoma in situ. It can be anywhere in the breast, and it's actually as likely to be in the breast where you found the lobular carcinoma in situ as in the other. So it's a marker for increased risk of breast cancer anywhere in the breast tissue. Margins are not meaningful. You can't just go in and excise lobular carcinoma in situ like you would typically do with ductal carcinoma in situ to get rid of that focus of risk. And it's a management dilemma because either you do nothing or you take away all the breast tissue. Those are really two, and it's a very radical thing, obviously, to do bilateral breast tissue. And for the most part, people don't do this. There can be overlap and mix between the two. Sometimes they coexist. And it could be that the lobular carcinoma in situ can migrate into the ductal system, and the DCIS can migrate the other way, and the two can intermingle. And there's a stain that's called E-cadherin that is very helpful if you're not sure what you're dealing with, or if you are both, and I'll show you a picture of that.
So
That's positive for ductal cells.
Does everybody understand? I mean, this sort of always confused me. It's associated with increased risk, but anywhere in the breast. So does the implications of that are what?
Genetics.
Genetics, or it's what I used to tell my parents when I said, you think I'm screwed up, it's either your genetics or it's your upbringing, right? So it's nature and nurture, but even for the cells. So if it's not the genetics, then it could be that whatever is setting off this abnormal growth is doing it for the whole, whether it's hormonal or carcinogens or whatever it is. It's not just that one local place, but it's everywhere. So it's sort of both.
We better move, and we're not going to get to the end. All right, so here's an example of a lobule that has been greatly expanded by lobular carcinoma in situ. And the rule is that, you can see what happened here is that the lobules no longer have any spaces in the center. The cells have proliferated, so they have crowded out those little spaces. And so if you had something that just had crowded out the spaces, but the size of the lobule was still the right one, then we call it lobular hyperplasia. But it's not just considered to be carcinoma in situ. But when you get the lobules expanded to this degree, then we call it lobular carcinoma in situ. And you can imagine that there is, a gray zone there too. You look at it and you say, is it really expanded or is it not? And sometimes it's hard to tell. So we often look around, and lobular carcinoma in situ is usually not one spot. It's usually several lobules that are, or many lobules that are involved in it. So we gauge from the whole process. Next. So it's common for it to be multifocal, meaning it's in many different areas. It's often bilateral. Is this my slide?
No, I think it was mine, though.
Yeah, I think so too. Doesn't look familiar.
So, but again, this is, as Britt was saying, it's a risk factor for invasive cancer. So once somebody is found to have LCIS, then you want to follow them more carefully. You may, as Britt was saying, it's hard to treat. Do you want to, treat aggressively,